Yes, coming to the definition, as I said before, it is the biochemical pathway which takes place mainly in the cytoplasm of our human cells, where glucose is broken down into pyruvate in aerobic condition and lactate in anaerobic condition, and finally it aims to produce the energy in the form of ATP, that is adenosine triphosphate. And it was discovered by three scientists, namely Emden, Meyerhof and Parnas. So obviously it is also called as EMP pathway. And there are mainly two phases of glycolysis. First one is preparatory phase and second one is payoff phase. And in preparatory phase is also called as energy investment phase, where ATP is invested during that phase, whereas ATP is generated during the second phase of payoff phase. And in preparatory phase mainly there are 5 reactions which comes under preparatory phase, where glucose is converted up to Glyceraldehyde-3-Phosphate. So that reactions mainly preparatory phase, where ATP is invested and ADP is consumed. And in payoff phase, from Glyceraldehyde-3-Phosphate to up to final product pyruvate through 5 reaction, and it is called as payoff, energy generating phase. That is, ATP is generated from ADP, adenosine diphosphate. Coming to the steps of glycolysis, there are mainly 10 main steps which is involved in glycolysis. First one is phosphorylation of glucose. Phosphorylation means the addition of phosphate group. So first one, the substrate is glucose, and that glucose is converted into glucose 6-phosphate with the help of hexokinase enzyme. And second one, glucose 6-phosphate converted into fructose 6-phosphate with the help of enzyme Phosphoglucose Isomerase. And this is the conversion of fructose 6-phosphate step. And next, again the phosphorylation of fructose 6-phosphate, where phosphate group is added to the fructose 6-phosphate. So fructose 6-phosphate converted into fructose 1,6-bisphosphate with the help of phosphofructokinase. And next one is cleavage. So cleavage in the sense that fructose 1,6-bisphosphate is cleaved into two main products, that is Glyceraldehyde-3-Phosphate and DHAP, dihydroxyacetone phosphate. And furtherly, it is converted into dihydroxyacetone phosphate converted into Glyceraldehyde-3-Phosphate, and that termed as interconversion. They are converting internally through Triose phosphate isomerase enzyme. And next one, Glyceraldehyde-3-Phosphate converted into 1,3-bisphosphoglycerate with the help of Glyceraldehyde-3-Phosphate Dehydrogenase. And next one, 1,3-bisphosphoglycerate converted into 3-phosphoglycerate. And finally, 3-phosphoglycerate converted into 2-phosphoglycerate, whereas phosphate is get shifted from third group to second group. So whereas 3-phosphoglycerate converted into 2-phosphoglycerate, and with the help of enzyme Phosphoglycerate Mutase. And finally, dehydration. Dehydration in the sense that second group of 2-phosphoglycerate converted into phosphoenol pyruvate. And finally, with the help of Pyruvate Kinase enzyme, phosphoenol pyruvate converted into pyruvate, which is the end product of glucose metabolism. And this pyruvate is mainly in aerobic condition, which is in the presence of air, glucose is converted into pyruvate, and that pyruvate it again converted into lactate with the help of lactate dehydrogenase enzyme. In the absence of air, anaerobic condition, the glucose is converted into lactate. That is, in the absence of air, it is converted into lactate, and in the presence of air, it is converted into pyruvate. So these are all the main 10 steps. So these 10 steps to be achieved for the glycolysis to be active in our body. If any one of the product or any one of the enzyme affects or deficiency of any one of the enzyme, which leads to carbohydrate disorders like diabetes mellitus, galactosemia, fructosemia, lactosemia. So if all the enzymes to be work properly for the conversion of glucose to end product of pyruvate.